DNA barcoding, unlike a lot of the technologies that we've heard about, started as a pure research tool. Biologists, actually a biologist in Canada about 10 years ago, had this idea, standing in a supermarket, that if you could tell the 40,000 items in a supermarket apart with just a 12-digit universal product code, there must be a part of the genome that would tell all species apart. There's about 2 million known species. And his research led him to a particular part of the genome. It's about 650 bases long, which is a really, really small piece of DNA. And it does work, unbelievable, to tell species apart, which means you can do very minimal biotechnology to get this gene sequence and then tell what anything is, what species it comes from, using just a microgram of tissue. Well, that's of course a very useful tool if you're doing taxonomic evolutionary biology research, but it also has implications for the real world. And the Google grant allows us to really make that jump from a research tool into the real world. And the project is about endangered species. There's about 2,000 critically endangered species that are protected from international exploitation, international trade, by a UN convention, the Convention on International Trade in Endangered Species. 2,000 that are prohibited from international trade, another 30,000 that are regulated. So how do you stop poachers and illegal traffickers when they're getting very, very smart, driven partly by organized crime, driven partly by terrorist organizations that are using trade in ivory, rhino horns, other rare animals and plants to fund illegal activities? Well, DNA research, DNA forensic research, just the sort that you see on crime scene investigators, can be used for wildlife crime prosecution, and that's what our project is about.
So it's like CSI wildlife?
Exactly.
Yeah.